Hello, everybody. So welcome to our first Science Communication Forum. So we will give the attendees 3 more minutes to see if we'll have a bigger quorum, and then we will start the webinar at about 4 p.m. So kindly be patient for 3 more minutes, and then we can start the webinar. So, hi everyone. I hope that everyone is doing fine. So to start you off, my name is Caroline Thuo. I work for the African Genetic Biocontrol Consortium, and I'm here on behalf of the Science Communication Forum, which is our first webinar, and I want to welcome you all officially to this webinar. And the objectives of this webinar, first of all, is to raise awareness on communication of genetic biocontrol technologies in Africa. And we also want to address the importance of community engagement and education in genetic biocontrol technologies in Africa. And under this, we are looking at some of the challenges some of the impacts, some of the gaps, and also recommendation in the communication of genetic biocontrol products. And at this point in time, please allow me to introduce our moderator for the day. So our moderator for the day will be Gloria Ogbaki, who is from Nigeria. And Gloria is the communications lead for the NBMA in Nigeria. So Gloria will take you through some of the plans that we have for the day, and also will introduce our first speaker, and later on our second speaker for the day. So I know as the webinar goes on, you might have questions. So kindly put your question on the Q&A or on the chat, and then we will be able to address them as we go along with this webinar. So welcome. And Gloria, please take over as the moderator of the day. Gloria? Okay, so as we wait for Gloria, you will allow me to introduce our first speaker for the day.
Okay.
Okay, good.
Okay. Hello, everyone. Hello, everyone. It's great to be here. This is our maiden edition of the Science Communication on Genetic Biocontrol. And we'll be having a series of these webinars. So I'll just like to read out the objectives. I don't know if Carol had taken the objectives. If not, I'll just like to read it out again. Number one is to raise awareness on communicating genetic biocontrol technologies in Africa, to address the importance of community engagement and education in genetic biocontrol technologies in Africa. This will address challenges, impacts, gaps, and recommendations in the communication of genetic biocontrol products. Okay, so I'd like to introduce our first speaker, and he is going to talk about the importance of community engagement in pathway for developing genetic biocontrol products. And that is Andrew Kigundu. Is a research officer in the National Agricultural Research Organization, Uganda. I would just like to say, okay, Dr. Andrew Kigundu is a principal research officer affiliated with the National Agricultural Research Organization in Uganda. He specializes in plant biotechnology, regulatory biosafety, stewardship, and communication for policy advocacy in Africa. He has served as a leader of the Biotechnology and Biodiversity Program, leading teams that worked towards sustainable utilization of plant genetic resistant cassava in Kenya. As CEO of Papyrus Technology Limited in Kampala, he is currently consulting for the Gene Convene Global Collaborative on developing communication tools for regulatory decision-making in Africa for gene drive technology being developed for mosquito control. That is a whole lot, and I know we are going to learn from him. So sit back as I give the floor, hand over to
Okay, thank you, Gloria. I would like to quickly share my screen, and yeah, host, please allow me to share my screen. Of course, please allow me my screen. You the host? Oh. Carol,
Yes, Andrew, we can hear you.
do you have host status? Can you please allow screen sharing?
Yes, I have.
Okay. It still says I'm not the host has disabled participant screen sharing.
Let me do that, Andrew.
Okay, now it's working. Now it's working.
Okay.
Now it's working.
Okay.
Now it's working. So can you see my screen? I presume yes, you can.
Yes, we can see your screen, Andrew.
I have a problem with double screens here, so I might not be able to use the slideshow. I think we can manage. I have used big fonts to be able to allow everybody to see what I'm speaking about. So thanks, Gloria, for the introduction, and thank you all for sparing the time to participate in our webinar. This is the first, as Gloria said, first webinar of a series of webinars that we will be conducting as the SCICOM, Africa Science for Communication Forum. And we are grateful to the African Genetic Biocontrol, PAMCA, and other participants, other well-wishers and our supporters, in particular, Jim Convene of FNIH. So it's an honor also for me to be the inaugural speaker of this webinar series. So I'm anxious to make sure it goes well and that the rest of the webinar series become a big success for our forum. I will speak to you about the importance of community engagement in the pathway for developing genetic biocontrol products. And I think that in future, this is going to be a continuous topic for discussion, and we will endeavor to invite speakers from projects around Africa that are already doing this type of community engagement. It's a bit new in the area of genetic technologies, and we think that many more projects that are fronting or that are developing genetically engineered tools and products in Africa and other parts of the world need to start to use community engagement during the development process, but also, as we will see in the slides I have, ensure that the projects run smoother to their end than it has been before. So, so this basically will be introductory in nature, and we will have opportunities to invite speakers who will go in depth in terms of practice, experiences, challenges, so that we learn more from each other. And those of you who have read around, or if you will start to read around, you will see that the methods and concepts around community engagement are varied. I guess it's because of the differences in projects, but also the ideas and the experiences have been varied. So community engagement or stakeholder engagement, as some people call it, is the process of identifying, analyzing, planning, and implementing actions to involve and influence eventually stakeholders for a common good. So we develop these projects to create changes or to develop new products, to develop new ways of doing things. But in the past, research has tended not to involve communities or stakeholders in a participatory manner. It is a key part of sustainable development and can help organizations consider the needs of everyone who has a stake in the organization and the project, fosters connections, trust, confidence, and buy-in for the key initiatives being developed, addresses expectations of stakeholders, prepares them for change, and offers insights into innovative processes, agenda of the organization and the project. The community engagement in research supports mutual respect for values and also strategies, and focuses on the processes and the results of the project. And some of these values most especially are environment, health, and other social well-being factors. And communities are basically the groups of people who live near enough to an experimental site or a laboratory or field trial where a release or particular piece of research is going to be undertaken, that and would have a tangible or immediate interest in the project or its outcomes. So organizations and projects usually would develop an engagement framework. And again, as I said earlier, these can vary a lot depending on the nature of the project. So an engagement framework would then commit this organization to be relevant, especially trustworthy, open, and responsive, as well as inclusive of as many stakeholders as possible. So it would involve defining the stakeholders, their roles in the project. And by the way, some of these stakeholders, as you can appreciate, would even be colleagues in the same lab, in the same building, in the same department, or in the same institution, would also be part of the stakeholders in the project. So it involves analyzing the stakeholders, their interests, their expectations, and their influence, and also plan, therefore, to engage with the stakeholders and communicate to them effectively. This reminds me a bit of on when we discuss communication efficiency and effective communication tools and why, and it becomes very important again to engage adequately as long as the appropriate stakeholders have been analyzed. So then implement the engagement plan and measures to be as effective. Information disclosure and sharing. This is where trust is built. Disclose as much information as possible and share feedback, share results with the stakeholders so that they continuously be part of developing the project. Also very important is to capture the feedback, and this can be used to improve communication or project activities, plans, and also project profiles. If you are developing a product, some of the profiling of the product might be information that you get back from stakeholders, names, languages, and different aspects that you may not have thought about if you do not engage the communities around the project. Also, there's grievance management, and this grievance management comes from sometimes issues of misunderstanding and issues of people feeling or community members feeling uninformed or just have questions. So it offers an opportunity for fulfilling unknowns that people might have, or sometimes, as some people have seen, the media or the anti-technology activists might bring in wrong information to try and divert the project. So within our frameworks, our engagement frameworks, we could provide room for managing such grievances from time to time. So why effective communication? We just remind ourselves of what it is really about, not only conveying message, but conveying the message in a way that other people can understand and respond. So it requires sending and receiving, and the sender delivers the message, and the receiver responds to the communication cycle. So this ensures smooth flow of ideas between both parties and prevents misunderstanding, which in many times such science projects encounter a lot. So it's about making sure that what you want to say is what you say, and is and it is what the audience interprets. So stand in there and make sure that as you communicate, you are also receiving response to those you're communicating to. Okay, community engagement has several levels, and these are strategic levels. So as a researcher, you inform the community of the project in a way that the project understands, or the community understands, and therefore you receive feedback from them. Communication goes really between stakeholders throughout the project. Community participates in activities such as if it's a mosquito project and you're collecting mosquito species in the community, get them to participate, look in the microscope and get them to understand what the mosquito life cycle is about and involve them as much as possible. So then what comes out is the community and researchers start to collaborate on every aspect of the project, on decision-making, and ownership is therefore shared. So this is what we look for. We look for the communities to share, to be part of the project, to contribute on decision-making. Perhaps they will even show you places that are better for you to do the work or to do the sampling than you might have previously thought. So involving them in all these aspects creates that collaborative effect that we're looking for. And therefore, the community starts to lead, starts to feel that the project and in collaboration with the researchers. And obviously, if this relationship is built, you can imagine that future of the project will be brighter, the impact will be better, and the uptake of the technologies when developed would be much faster. So a list of the benefits here, as we have seen earlier, building trust. A lot of the communication sometimes you see in the media reflect scientists as people who are not trustworthy, doing all kinds of ungodly work. But if communities are engaged, what you do, why you do it, I think that the trust in the community and the overall stakeholder can improve. Also improve research methods. There's a lot of, especially field research methods that are improved when you engage the community. That makes the research more relevant, enhancing collaborative partnerships, improving quality and impact of the science and research, enhancing participant recruitment and retention, therefore driving social transformation. I think that we would be building, social scientists will be building community science, so to say. It creates opportunities to improve and consent, improve the consent and consent on the processes, and identify and resolve pitfalls, enhancing the amount of public involvement and contribution, communication that encourages involvement, fosters adoption of new technologies, reduces anti-technology criticism from activists, and also improves regulatory outcomes. I, in the past, have experienced regulators who rejected projects simply because they had not done enough community engagement. And also, as some of you may already know, the Cartagena Protocol Article 23 requires public awareness and participation in the regulatory assessment review of applications for products of genetic modification. So this article requires projects to promote and facilitate public awareness, education, and participation concerning the safe transfer, handling, and use of living modified organisms, and the other subsections especially refer that country, the parties to the protocol, to the endeavor to ensure that public awareness and education is conducted, and that all parties in accordance with their respective laws and regulations should consult the public on the decision-making process. So you will see that most countries have a public participation in the process of review of GE projects. And so if this is at a time of reviewing your project for approval, but if you have done this right from the beginning with the communities, so you can see that you're building towards improving regulatory review and acceptance or approval of an application. So in the next couple of slides, just showing you how one of the genetic biocontrol projects in Africa, Target Malaria, engaged the public. They have developed policies on stakeholder engagement, on seeking approval, on seeking consent and agreement to conduct project activities, either on them as individual persons or in their homes as a family or in the community. They have engaged at those different levels and continuously throughout the project. At every stage of the project, they would get back to the community and engage them, providing information and seeking feedback. When they receive this feedback, you can see the project staff there has a tablet and is collecting data responses from the participants of this particular engagement activity. So these responses and the engagement activities themselves form part of the accountability, form part of the reports moving forward to guide the project, but also to ensure that as the project goes on, there is adequate information for the public to engage. This is another one where groups, much bigger groups, where agreements are sought from the community as to when and how projects or project activities would be carried out in these areas and the areas of operation. So I invite you to visit www.targetmalaria.org, and a lot of this information has been assembled there for you to appreciate how at least one of these projects or one of the genetic biocontrol projects has addressed the issue of public engagement in research and development. That's all I had. Thank you, Chair, and thank you all for your audience.
Okay, thank you so much. Thank you so much, Andrew, for that very comprehensive talk on the need, the importance of community engagement in the bid to develop genetic biocontrol products. All right, so the floor is open now for questions. The floor is open now for questions. But I have a question before we move on. I'd like to ask, at what point does the process of engagement start? At what point in products, at what so that at what point the community needs to be carried along?
Yeah, Gloria, if I may answer, other people can chip in if time allows. But I think obviously, as you know, research starts from the very, very basic. But once you have proof of concept that a technology would have value in the environment, would have value as in commerce or in any other as a public product, I think this is when you would want to do the uptake research, the field research, field trials, for example, to show that yes, the product is efficacious, the product has value. I think that's the right time. You get out there and engage the public both in the areas close to where you're doing the research, but of course, gradually as as science communicators, we should be able to gradually get this information further and further along other stakeholders that would have interest, but also would be key participants in decision making for this project. So at the time of proof of concept as complete, you want to go out and confirm in the environment and do environmental releases, limited environmental releases, to collect more data is when the communities need to be engaged.
Okay, thank you so much. So I have another question here. It says, can a community that has agreed to a project backtrack? What happens in such a case? I don't know if you got that.
Yes, I got that. Thank you for
Yes.
that, John. I think that these are some of the challenges which I didn't include here because my talk was really to, just to break the ice and to get the discussion going. But some projects obviously have received such challenges where communities again influenced by external forces tended to want to backtrack from earlier agreements they had. But I think that once the engagement, or if the engagement is done properly and involves as many stakeholders as possible, this should be very limited. But hey, I'll keep that question also and build more ideas around it. Or if anybody else in the audience would give us an answer, you're welcome.
Okay, thank you. So the floor is still open for question. The floor is still open for question. Thank you so much, Andrew Kigundu, for your presentation. All right. I don't know if there are further questions. If there are none, we'll have to move on to the next presentation. But I must say it was a very good presentation and I really, really loved it. And I know so many people also loved it. I don't know if there are any other questions. Thank you so much, Andrew, at this point, for your presentation. We'll move to the next presentation, and that is on the motivations and methods of engagement in genetic biocontrol technologies in Africa and the need for evidence and genetic biocontrol technologies in Africa. Okay, so I'd like to invite Dr. Murenga Mwamali. He is an agricultural development, seed systems, crop improvement professional. I'd just like to say one or two things about him before he starts. Dr. Mwamali is an experienced agricultural development, seed systems, crop improvement professional with a demonstrated history of working in the National Agricultural Research System, CARO, management for 15 years. Skilled in project management, coordinator of the Water Efficient Maize for Africa, WEMA project, which was up to 2019. Donor relations, working with AATF, Feed the Future and World Bank projects in CARO and seed companies. Value chain management, food security and crop improvement, climate smart agricultural technologies on drought, diseases and pests, capacity building, strong leadership and advocacy, experience in development, management and commercialization of technologies and hybrid needs, community and social services. You're welcome. Dr. Mwamali, you have the floor.
Good afternoon, everyone. I don't know if I'm visible and audible also at the same time.
Yes, you are audible and we can see your slides.
And myself?
Yes, we can see you also.
Yeah, so let me put this in the slideshow mode. First of all, I would like to thank the organizers of this meeting for inviting me as a speaker today in the science communication on genetic biocontrols. I'm very glad to be here and to be among the first of the speakers in this series of webinars on science communication. I'll be talking to you about motivations and methods in community engagement and the need for evidence in genetic biocontrol technologies in Africa. That is my name, and I'll go straight into the subject of the day. The question we have is, what are gene drives? My little understanding is that gene drives, what are gene drives? A gene drive is a gene which rapidly spreads from one generation to another generation without following the Mendelian inheritance laws. As you can see on the left side of the figure there is that on the right side, on the left side, we have the normal inheritance, and on the right side of my screen is the gene drive inheritance. And the essence is that the gene will move. I have tried to put it in a little more easy definition without going into the depth of science. But basically is that the gene will spread from generation to another without following those laws of inheriting half of the maternal and half of the paternal. So why gene drives? I'll mention to you a few proposed applications in human health, in the environment, and agriculture. And at the same time, as you notice that we have gene drives having a cross-cutting role in the three sectors of life, then the human health, the environment, and agriculture. Specifically, in health, you will see that we are aiming at immunizing animal resources, animal reservoirs of diseases, controlling the vector-borne diseases, for example, what we are going to be talking to you about. Assisting in threatened species, having sustainable pest management tools, and also developing non-toxic herbicides and pesticides. For the purpose of this presentation, I will basically be looking at GM mosquito, the gene drive mosquito. As has been mentioned earlier, by my early speaker, Andrew, is that there are various pathways to deployment of gene drive products. And this starts from the laboratory studies, moving on up to small-scale releases, large-scale releases, and even beyond. If it was a product that we are, for example, is a food product, we'll be talking about placing it on the market and carrying out post-release monitoring and evaluation of that, and having in place all the safety guidelines that are required from the lab, the compliance, the regulatory compliance in terms of at the national level, first at the institutional level, at the national level, and all the way up to when the product is moved to the market. Now, next is sorry, these slides are not moving. Can you hear me, please? Hello?
Yes, we can hear you.
Yeah, the slide, okay. Yeah, so I was talking to you about beyond the pathways to deployment, we now have we have to maintain integrity. the integrity of this product. Now in the space that we are discussing is GM mosquito. It requires that we have considerations on stewardship, in which case we have to analyze the obstacles and constraints that may hinder uptake. We have to understand that we have to ensure the marketing and deployment of approvals are received in timely manners in the target countries of deployment, because now for gene-derived mosquitoes, it's generally going to be, the targets will be the malaria endemic countries in Africa. Then again, we have to have strategies that support the stewardship process and access to the technology in those malaria endemic countries. Other issues of concerns within the stewardship space would include the intellectual property rights, the need for monitoring of the effectiveness of the technology in the target areas, and that there must be an assurance of full compliance into the law at the national, international protocols like the Cartagena Protocol and other protocols that guide research in respect of gene drives and or GM. So I'm talking to you, I'm posing a question here, which is asking that, what is motivation? Now, the motivation is the driving force behind why we are doing this research on gene drives. And it's a function of the expected successes and the value that will come from it. The motivation for gene drives is generally guided on 3 levels: that we have to have the activation, why we want to have it. We must have the technology that is actually going to be available, and that it's intense. So whom do we involve in the gene drives? Who are our stakeholders? Andrew has mentioned a few, and I just want to put it in a manner that a stakeholder is a person, group, or organization that have particular interest in a policy or a program. In this case, the gene drive. This will involve government agencies, the health institutions, both at the national level, local and county level or something. It will also involve policy makers, the laws makers at all levels of government, be it the national government or devolved governments, regional or other. Then there are the funding agencies and other beneficiaries. We also have to make considerations of civil society, the faith-based organizations, and service providers. For example, when we talk about service providers for gene drive, who are the people or institutions going to be involved in the production of these mosquitoes en masse? As we move from the lab and move to post-release and placing the product into the environments of people live, we will need monitoring and evaluations. That will be in line with the laws and the regulatory institutions. Then what is the motivation? Why the motivation to have gene drives? We have 3 levels of that consideration, that we must have communities and stakeholders must have the knowledge. They must understand the complexities that are involved at all levels of social, political, economic, and ecology, in which these gene drives are going to be deployed. We must also understand that the motivation would be that people or communities must understand the principles of justice and demand for the transparency, and there should be a well-informed consent about these experiments and even future experiments, and how that will affect the people and their landscapes. And the third level of why the motivation for gene drives is that the engagement of the communities it creates opportunities for mutual understanding and trust among the diverse groups of people. However, it also presents, the engagement presents challenges that must be recognized and addressed throughout, from the lab all the way to post-release of the given gene drive. Oh, next slide. Sorry if my computer is not behaving well. Oops, Carol, anything you can do to help? Hello.
Let me see if I can project from my side. And then we go to that page.
No, it's fine. I don't know why it's giving me breaks that I don't need. So when we're looking at stakeholder engagement in gene drives, we must be cognizant of the fact that we have a diversity of people, from those who are totally unaware of gene drives, those who will be aware, but they will choose to resist, those who are neutral, and others will be supportive, and others will take the lead in terms of, I'm looking at a scale, a space where we are having awareness that is increasing from those who are unaware to those who are the innovators. So there is a need to engage all these stakeholders at all levels, and the message packaging must be in terms of understanding. No, once you know that this stakeholder group is in the unaware, the messaging will be packaged in a way that it can fit what their interests are as we carry them along. So much has been talked about community engagement, and I just want to reiterate that it is a process of working with a group or groups of people on a shared or common value. There are various methods of community engagement in gene drives, and these methods that are available depend on, one, the goal for which the project is about, the context in which the project is operating, and the levels of engagement that are required. Like I've mentioned before, of from the laggards to the innovators. The community engagement method that is deployed must inform participants or community fully about the purpose of the project, and that the engagement must maximize the opportunities and encourage interaction and exchange of information between and among the community membership, including the research team. So while thinking about methods of community engagement, there are two levels of community engagement methods. One is the direct engagement, for which will involve town hall meetings or community gatherings or forums. In East Africa, we call them barazas. In the west of Africa, I don't know which particular country, but they are called durbas. I don't know if my pronunciation is correct. However, the basic point here is that these are face-to-face interactions between the research people, the research team, and the community that is involved in them. The method of face-to-face interaction has one advantage, that there is an advantage of delivering information firsthand from the researcher to the community stakeholder group. However, the method of community gatherings has also its downside. One is that, how do you identify the relevant communities that are going to be engaged? It's also challenging that you may not be able to discern how people have understood the information that is being shared, especially when the community, those meetings are have very large numbers of people attending. And also, there is a tendency in this kind of forums to have certain groups of people who talk too much in meetings that it masks the ideas of the others. And it sometimes fails to capture the perspectives of those members that may not have attended that meeting, that is the community gathering. Those the absentee members may not necessarily capture their perspective of the project. Another method that is direct in terms of community engagement is the focus group discussions, in which you, as a research person, have identified, say, for example, vulnerable, marginalized groups, women groups, chiefs, opinion leaders, and community development officers, activists, patient groups, groups that are affected by the technology. It is easy to carry out focus group discussions because the management of such kind of groups are easy. However, the bigger picture of the community gets missed out because the biases that these groups can come with may not necessarily be community representation. There are also indirect methods of community engagement in gene drives. One is that we get to have engagement with communities through representatives of organizations that are non-monetary, people who have a common interest coming together. They hold regular meetings and they are organized in their decision making. This has the advantage is that we are able to capture a wide range of stakeholders, including government officials and faith-based organizations and other stakeholders that are within the representation bracket. And the other advantage of this methodology of engagement of community through representatives is that the possibility of long-term partnership between the group, the research group, and the community is high. However, there is a challenge with engagement through representation. How do you identify the people who will commit their time throughout the project life? Two, the membership of these representative groups. Who are the best people to serve as members of this? In that case, then, it calls for researchers to have what I call must do. They must do for the research teams in gene drive projects, that they must be accountable. Accountability is one best practice that must accompany the gene drive research, such that they inform, they educate, and empower communities. By that, they'll also have to make sure that the community also understands their own role. The risk that the scientists come with is what we call inherent biases. The research team arrives at a target community and they have their biases they come with their biases, in which case we lose objectivity. So researchers must avoid their biases as they come over into engaging the community. However, while we engage community, there is no single strategy of assessing the effectiveness of these community engagement methods. It all depends on the nature of that research and the goals of engagement. A hybrid of methods is required to provide information, to consult the community leaders and their representatives, and actively involve them at various levels of the community membership. While all this is going on, there is a need for evidence that these gene biocontrol technologies are actually working in Africa. the value of the genetic biocontrol technologies in Africa will generally depend strongly on its potential public health contribution. Two, the potential risks and the activities and resources that will be involved in the implementation of the gene drive technology. Despite the potential advantages of gene drive technologies in Africa, much of it remains theoretical. And it's until the field trials demonstrate the epidemiological efficacy and the entomological efficacy of these gene drive mosquitoes. At the same time, it has to capture issues to do with safety to the human. We must carry out the safety evaluations and the efficacy. There must be regulatory and ethical approvals that would capture biosafety and social acceptance. The other thing is that the characteristics of persistence and spread of this gene drive should increase the suitability of self-sustaining gene drive mosquitoes. That should be applicable within the very many diverse ecologies in Africa, including the urban environments and some of the resource challenged persons and people conditions where maybe how do the poor access this kind of technology? Is the technology going to be persistent and spread post-release, even and continue working for the people, even in some of the challenging and disruptive conditions? Disrupted conditions here will mean countries, for example, that are facing civil strife, and it's not from it's all over in Africa. To conclude, and maybe mention a way forward here, is that there is a need to identify effective strategies to engage the, sorry about this, effective strategies that will, there is a need to engage, to identify effective strategies to engage research participants and communities beyond the sampling stage. There's a need also to explore the broad consent that as a community you say, we agree. As you take samples beyond the project, what will you be doing in the samples? Those information must be included in the broad consent by the people and community. And also, finally, there is a need to design and review continuously the strategies of community engagement and the guidelines that relate to the gene drive studies. Thank you very much for listening to me. Hello?
Okay, thank you, Mimali, for that presentation. It was indeed a very informative presentation. So I'm seeing some comments on the Q&A. So we have, yes, true for effective communication, great talk. That is Samira. We also have Amelie has a question. I think it was answered, but she says another comment, I agree. Rigorous regulations are a must. So can you please expand on the current status quo with regards to Africa? I bet that's a question for Mimali.
This question is a good one. However, I don't have the fullest of the answers, and I beg that if by chance I miss out on mentioning what may be useful, I'll be happy, because on the participants I see my seniors there also. The situation on status quo in Africa it's not a general one, but while we are looking at gene drives, one of the most important things that is going to help us is the presence of political will. We have learned that political will can actually accelerate technology take up faster than just having laws, implementing them fail. I mean, having good laws and not implementing is a very big challenge in Africa. I'm borrowing this from the science that I am involved in on genetic modification for maize and BT maize in Kenya, that when we have good political will, there is a very big advantage in terms of quick uptake. For example, Kenya, for example, if I may, you allow me to use that example, but the current government is very supportive of genetic modification uptake. And we see institutions at even from the national level at the county levels, already institutions are working on accommodating the cabinet decisions on uptake of GM maize and GM cotton and GM cassava. Now, if we have an understanding, by not only an understanding, but the acknowledgement that our country or a specific country in Africa is endemic, is malaria endemic, then and we have goodwill from the political side, then the uptake for this technology is much faster because the management of countries will already be understanding that here we have a solution that doesn't have to continue sitting on the shelf, but it's useful and it's going to save populations against malaria, for example. I beg to stop there for now.
Okay, thank you so much. Thank you so much for your presentation. It was a very good one, a very good topic. And talking about motivations and methods of engagement in genetic biocontrol technologies in Africa and the need for evidence in genetic biocontrol technologies. And some very key points that I was able to pick is that communities must demand transparency, meaning they must be carried along. And the fact that accountability is a best practice that must accompany gene drives. And I really, I really want to key with you on the importance of carrying the community along. It's very key to have a successful engagement. Okay, so right now we have some questions already. And I'm seeing one that is saying, okay, it says, who should lead or engage the stewardship? Is it the same as a business strategy for products? I don't know if that question is meant for you or for Andrew. I don't know. Did you get that question?
I want to make, I guess we want to make a comment on who should take lead.
Yes. Who should lead or engage the stewardship? Is it the same as a business strategy for the product? So I think this person is trying to talk about who should lead the engagement.
Yep, I got it.
Yeah.
Mm-hmm. Shall I go or shall I?
Okay, let me just take the next question I have here. It says on the current status quo with regards rigorous regulations. What is the status quo as regards to Africa? I think those are the two questions we have for now.
I didn't get the second when there was a break.
Okay, he said, okay, let me just read that in full. It says rigorous regulations are a must. So can you please expand on the current status quo with regards to Africa? This person is trying to, her question is around regulations when it comes to gene drives. What is the status quo of gene drive regulations in Africa?
My quick comment is about the stewardship. Who should lead? The question, the leadership of stewardship is not embedded in a particular space where you say it's the researcher, it's the community. It's supposed to be a wholesome package, because if we may parallel this from the understanding of, say, BT maize, right? That you cannot have BT maize deployed into the market and assume that once you have it in the market, it is already functioning. There is a risk of breakdown of resistance. So there must be some refugia programs. And the refugia programs would mean that you have to maintain a certain level such that resistance to BT is delayed. And this will involve both the researchers and the users of the product. Parallelly to that, on gene drives, I said that we have to engage communities, and we're engaging communities coming from the understanding that first, that the product, that the gene drive product is safe, that we have already carried out the safety and efficacy evaluations, and that the regulatory and ethical approvals are already in place, and there is social acceptance. Now, when we deploy gene drive products into the market, if the three levels of safety, evaluations, efficacy, regulatory, ethical issues, social acceptance is done, then it becomes easy to have the products deployed into endemic areas, and people should see value. When people see value in the technology that is being deployed, then they will actually accept it and they'll be able to keep up with it. At the same time, the engagement of regulatory institutions in, like I said, the service providers and the implementers, is that if these institutions are going to be producing mass gene drive mosquitoes, it is incumbent upon the regulatory institutions to make sure, like in Kenya, for example, it will be the National Biosafety Authority, to make sure that the product that is going out, remember now we are talking about a live product, which is a mosquito going out, that the integrity of this insect and that the presence of the gene that is being inserted is efficacious enough and persistent. It is the institutions that are at regulatory level that are it is within their mandate. And the other question on the status of regulations on gene drives, I am not very sure whether all countries within Africa have. I'm not so sure, but what I'm very aware of is that I will not want to speak for Nigeria or any other, but I'm aware that within Kenya, we have the gene drive, gene editing regulations, and GM biosafety laws. All these laws within all the national, what we call the national biosafety organizations, authorities, there is, if you are to do a gene drive mapping of laws that are within the African space, there are very few countries that have those laws, and very few of them have even the trials, the ongoing trials on gene drive mosquitoes. So I leave it at that for now.
Let me attempt to give some response to that, Gloria.
Okay. Oh, yeah, go ahead, please.
I will quickly share a screen where I will show a quick map of an analysis we recently did to show which countries had some form of a regulatory mechanism for genetically engineered products. And we believe, even if these products were initially plants, we believe these would still be the same laws and regulations that these countries would use, adopt them to address issues for gene drives, gene editing, synthetic biology, and the others. So if you can see the map, can you?
Yeah.
This is the map we've developed. It's on Gene Convene website. VI, if you go there, you will see it's interactive. So all the shaded countries on this map actually have some form of framework for which they can use to make decisions on genetically engineered products. Which is quite a number, you know. It looks, Africa is quite busy with the regulations, actually. Only that the functionality of the regulations is dependent on so many other factors within country that, you know, becomes a bit difficult to analyze deeper. But you see that if you scroll or if you hover the map, the pointer, you will see the name of the country. So all the green ones actually have experience in reviewing applications and therefore have conducted some open field trials of GE crops in their environment. The orange ones, the brown ones have laws, but they have never used them for reviewing any application. But you can see that certainly, yes, there's quite a number, and I'm happy that Africa has made this progress. For gene editing, 3 countries, which is Nigeria, Ghana, Kenya, have already published gene editing guidelines and regulations. So these are even more advanced than the European Union, because the European Union still hasn't made up their mind on how they are really going to treat gene editing. The little arrows here, the lines are showing the countries in which 3 major projects are working on mosquito genetic biocontrol of mosquitoes for malaria. Target Malaria is working in Burkina Faso, in Ghana, and in Uganda. There is a project by University of California working in Sao Tome and Principe. And there is a project called Transmission Zero that is working in Tanzania. I think that is a quick summary of the situation in Africa. As I said, these are all analyzed based on crops, but we believe this is sufficient, or the regulations in place can easily be adopted to regulate even mosquitoes, which has already been done in these other countries anyway. It means that all the other countries that have regulations, functional ones and non-functional ones, are in a prepared state to receive or to review applications on this technology. Thank you.
Yes, thank you so much, Andrew, for that map, because I was just about to say that Nigeria has we have a very functional regulation when it comes to gene editing. So thank you so much. Okay, we have another question here. He said, how to erase prejudice in the minds of communities about genetics before getting them to engage? I don't know if Dr. Morenga, he got that question. I don't know.
Yes,
I myself, I don't know if you get that question.
Yes, if I may try and address that, you're saying how
Okay.
do we remove community prejudices? That is a very difficult question. While the project will set up objectives, it is incumbent upon the research teams to be able to identify what we call you do stakeholder mapping, and that will also mean that you identify sites that are your target sites, but you want to engage a group after educating them. For example, you have to make sure that these groups of people, when you do a SWOT analysis on a stakeholder group and sites, where so that you situate. I'll give a simple example of what I've seen in Kenya, for example. We have a project that is with a pig company, a company that sells a lot of pigs, and I mean they process pig products from it, for meat and sausages and the likes. But you see, you cannot situate such a, you do all the mathematics and all the benefits analysis and cost effectiveness, cost benefit. There are other factors. For example, religion will not allow you to engage such kind of a big project in a Muslim background. So it is upon the research community to basically do a stakeholder mapping to identify areas and groups of people who to engage with, educate them, inform them, and be able to work with them. So for me, it starts with the community, with the research people when they are doing what we call stakeholder mapping. Because as I said, beyond the safety, beyond the efficacy, beyond the regulatory approvals, there must be the ethical issues, there must be social acceptance. So if those 4 labels are complied with, then the possibility of success of a gene drive project is high. Similarly, with what we have seen with GM products, not only in Africa and also elsewhere. That's my quick take. But my comment is open to more comments from the floor.
Yeah, thank you so much. Thank you so much. Okay, I think we have a hand that has been raised. Emmanuel?
Yes, can you hear me?
Yes, we can hear you.
Okay, so I think I had 2 questions, but I think the first one had to do with the first presenter, but unfortunately you didn't see the hand. I don't know whether I can put the 2 together. But just the second question has to do with a question the facilitator was trying to address. In Ghana, so actually I work with a media house. Now, in Ghana, the word GM or GMO alone is a problem. When GMO came up, I mean, the way the country received it or the public received it was not too good, because there were people who had come in, or let's say some anti-GMO people, and the publicity they did had actually overshadowed what the scientists were talking about. Currently, people do not just want to hear the word GM or GMO or genetically modified, and then any word you want to add to it. So the question I'm trying to ask is this: can there be a different model? Like, can there be a different way of packaging the same thing so that people can accept it? Because the first word of, or the first hearing of GM or GMO becomes a problem. And people do not want to even continue to listen to the content that you want to present, even though, per what I'm hearing, I mean, modifying mosquitoes to deal with malaria is a good thing for Ghana because there is a lot of malaria cases in Ghana. So how do we repackage some of these things so that the people can accept and adapt? And I have that link it to the first presentation because I overheard him talk about building trust. The trust of this had already been lost on the people. How do we repackage this so that the people can accept it? Again, in Ghana, a lot of people listen to the radio station and television station, but especially radio station, where you do not even need to mobilize people and go and do a presentation to. Sit on the radio, talk about it, and let people buy into it. How do we build a trust between the scientists and the media houses so that they are able to do the communication for the scientists? Because the very way you sit on radio and talk about GMO, and then a media person sits on radio and say, as for this thing, I am not sure about it. Then it goes into the audience and it's finished. So how do we build a trust between the scientists and the media, trust between the scientists and the people, so that whatever it is that the scientists are producing, the trust will be there within the people. It can be communicated right through the media to the people. And then again, can we repackage this whole thing back again to the people because the start was bad?
Okay, you might as well ask your second question for the first presenter so that he can take notes as soon as
So I've just tried to box the two in the
Okay.
same presentation that there is an issue of building trust. So how do we build a trust between the scientists and the media? It was within the first presentation. And the trust I am talking about is a trust between the scientists and then the media, trust between the scientists and the people, so that whatever it is the scientists are dealing with, the people can actually buy into it. The second one is, how do we repackage this whole thing about genetic modified or anything that, you see, when the people hear anything genetically modified organism, you know, then there is hyper within the system. I mean, they are coming again. What is it that they are coming to say? Because at the beginning of GMO, the communication didn't go well. Some people had already come to produce the minds of the people that genetically modified things are bad for the people. It changes the originality of the things. And therefore, these people are trying to temper with originality of what God has done. This has already sunk into the minds of the people. How do we repackage it? How do we then also get a trust of the people through the media so that these things can actually work?
Okay, so thank you. So I would like I will invite Andrew to start off answering the first part while Dr. Murenga takes over the second part. Thank you.
Yeah, thank you, Emmanuel. That's a very passionate question. We have engaged, we have thought through this considerably. But for media, I think that what other places have done or what we have been doing really is to build media friendship. Obviously, you know, media is also highly categorized. Some are investigative in nature, others are for sports, all kinds of categories. So we have started to build media individuals as friends of science, such as in this Science Communication Forum, we have membership of generally science journalism, or science journalists and media personalities that we try to bring together and build friendship and show them what we do and use them to package and deliver the promises of our technologies. And you are welcome to join and to bring in even more people that are, they have to be biased towards communicating development, science, agriculture, health, and things like that, especially where a lot of this new technologies is being applied currently. To go a little bit on your question about repackaging the term genetically modified organisms, I think it's going to be tough because we do not want to be seen as trying to avoid what the thing really is. What the US has done is they have replaced in their regulatory documents and many other communication material to actually use the word genetically engineered organisms or genetically engineered products. That was an attempt to actually be very specific on what they're actually making reference to, because genetic modification is too wide. It's a misnomer in terms of really the practice of inserting genes artificially, because even common breeding or normal breeding could also refer to genetic modification. And actually, that's why a lot of confusion prevails as to what is exactly genetically modified and what is not, because even humans, by the virtue of being products of sexual reproduction, we are modifications of our parents. So I think they're trying to use other terms, which I have seen people trying to do, especially in the health sector, the vaccines and others are trying to avoid the term GMO, but that's what it really is. And I think that with time, we will just change the previous bad mindset or bad image.
Thank you, Andrew, for that. I would like to make some quick comments on what Emmanuel just said. As it comes to developing different approaches and messaging, I think it's more about when people see maybe the scientists have first of all failed and allowed misinformation, disinformation, and malinformation to go ahead. And you see, the way the anti-technology activists package the information is in a way that it's emotive. And issues that are emotive are to do with the heart, and people will they will not care what you're telling them if they don't see, if it touches on them in terms of their feelings, they will be very careful not to take up certain things. But I also agree that it will take time. But at the same time, we need to increase our speed as the media and research to find a way of packaging this information, to find ways to package this information that is about these technologies in a more friendly manner and simplified manner, avoiding jargon and such kind of things. Coming back to your other way of part of the things that I want to share with you, with the listeners, I mean, the membership on the forum, are that in Kenya, to ensure uptake, is that on uptake on BT maize, for example, and and cassava, virus-resistant cassava, and BT cotton. We, as a team leader of that project, we carried out what we call seeing is believing tours, where different stakeholders are taken to different sites where we have the demos on this crop. And when they come over, the different stakeholders that I mentioned in my presentation, you have a message packaged for the religious groups. You have a message packaged for the political class. You have a message packaged for women groups, men, different stakeholder maps. groups, such that if this were to happen, and if it will happen, and it's happening, the stakeholders, they really don't care about what you say. They care about what is in it in terms of demonstrated value, demonstrated benefits of this experiment. And when they see value in what you're saying, which is directly accruing to them, the probability that they will take up the technology is high. We have seen farmers asking us for seed of BT maize while in the confined field trial, because they are saying, this maize is so good, I need the seed now. But you see, we were at the efficacy level, efficacy test, proof of concept stage. A similar thing with gene drives. If the stakeholders in the malaria endemic sites see benefits, demonstrated benefits, then the probability that they will take it up is high. I beg to stop there.
Thank you so much, Kigundo and Murenga. Okay, we still have more questions coming up. This one says, given that we are working on mosquitoes that can end up in locations beyond a release site or community, how far in terms of community coverage, the communication and engagement activity should be done by GDM developers or researchers? Okay, he's trying to find out who should do the community engagement activity. Is it the developer? Is it the researcher? Don't know if you got that question. And another one, okay. Another one says, what's the difference between gene editing and gene modification? Will gene editing need its own regulation from gene modification regulation? That's the question. And this other question is saying, is there any specific law which will be used to regulate gene drive? I think that has been answered, where Andrew provided a map to show. So please, we could give answer to the other two questions.
Andrew? Andrew?
Yes, sorry, I missed that. Can I have it again?
Okay. He said, given that we are working on mosquitoes that can end up in locations beyond a release site or community, how far in terms of community coverage, the communication and engagement activities, should it be done by the developer or the researcher? By the GDM developer / researcher.
Yeah. I think that current releases have tried to go into areas where the spread will not go beyond certain boundaries. They've tended to use islands, especially for trials. But my understanding is that once the project's finished and the product is supposed to be released, the whole community where the releases will be and the extent to which the mosquitoes will migrate would have to be involved. Yeah. So yes, so then that means then the community engagement would have to take care of the coverage as indicated by the migration potential of these mosquitoes, as well as the intended benefits of reducing their population and reducing malaria.
Okay, thank you so much. So I'm seeing a hand up from Samuel.
Yeah. Hi,
yes,
Gloria. Good afternoon.
Good afternoon.
Yes, just a quick one to Emmanuel's, a quick intervention to Emmanuel's question. In Ghana, like he said, we faced some challenges when we started the processes, and even today we were in court. The case has been adjourned once again between the matters of NBA and then food sovereignty. So we're actually working on it. Apart from that one, so that is just a follow up to whatever he said. The place of the antis in trying to sabotage whatever work we are doing. On the other hand, our engagement with the journalists. One, we are working with the Ghana Journalists Association, through the vice president, to coordinate a number of science journalists in this regard, so that we can have a media call, a press standing call, that as and when necessary, we can fall on them and then do the propagation. And then, like he may be aware, we met, I think about 3 weeks ago in a workshop where we invited about 20 journalists from the regions for 2 days on genome editing training. So we've started, the National Biosafety Authority, we've started working on some of these things to address some of these peculiar challenges that we face. So we want to assure him that we are not setting down on concern. With time, we'll bring all journalists on board. We are first starting with the science journalists, who we believe will have a firm grasp of whatever we are doing. And then from there, we'll reach out to the larger community. Thank you.
Okay, thank you so much, Samuel, for your comment and for that bit of clarification. I would like to hand the floor over to Carol to take it off from here. Carol?
Okay, so thank you, Gloria. So before we wrap this up, I've seen question from Aruna. It's saying, I wanted to know what we can do about activists who, whatever we say, manage to influence the population with false messages, and often they manage to have high-level research in their ranks. Is it possible to approach them, or does everyone have to stay in their role? I think he's trying to mean as scientists, a message may be put out there, but by the time it gets to the journalists and then to the population, then it has been diluted, the misinformation and the disinformation. So how is it that this can be dealt with? Andrew, Murenga, anyone can go?
My quick take, sorry.
My quick response to this is that, especially after having been in these fights for so long, is not to just keep doing what we are doing. Because misinformation, unless they have attacked you directly, how much effort will you have to do? And I think they do it on purpose to distract and delay and prevent you from doing what you should be doing. I think that unless it's direct and somebody has approached you for a response, you respond, give the correct information, and that's it. You really can't fight it, because there's big efforts behind it, and they do it on purpose. If somebody genuinely is lacking information, you provide it, and if they are reasonable enough, they take it. If they are not, and they are doing it on purpose, they will never. Yeah.
Thank you, Andrew. Probably I'll just a similar statement from my side and the experience I've gained over the years here in Kenya. That the purpose of the gene drive activity or GM, it's not about being confrontational. because being confrontational may not necessarily yield much, but is to have proper channels of communication in terms of as government institutions and those involved within the government, is to find ways. You know, people trust government quite, and if we can have a way of information from the research side going to the public through government mechanisms, the probability that it will be taken up as true is much more likely than just being confrontational with the activists, because activists are out there. One of them told me, "This is a job like yours." So you have to be just stay focused in what you're doing.
Okay, thank you for those responses. And also to add on to that, it had been mentioned before about building trust. So I feel that would also really be important when you're reporting or putting out your work or your message as a scientist, so that you can be you report to safe people as well. So during the last workshop we had, there was a point that came up from the communicators to the scientists. As you're communicating, do your due diligence, your research, so that you can know who is this person that you're talking to at the end of the day, so that when they go out there, they will not dilute your message. And then, so moving on, there's also another question that asks, despite the presence of biosecurity institutions, communities are still afraid to get involved. So what is the effective strategy that we can develop for better control of this technology? So by this technology, I think it's GMO or gene drive technology. That's what she means.
Andrew?
Yeah, so the question first starts off with the regulation, right? Yeah,
yes.
I think that we have not communicated regulation very well. In fact, recently when I was developing and looking at regulatory frameworks, I realized how little people know about the processes of regulation. Because people always say, "Oh, the GMOs is everywhere," but it isn't. It hasn't been approved, and then, but what does it really mean to approve? And who does it? Where is it done? And how is it done? I think this is also information that is lacking out there. To create the trust, as we just spoke about, we need to also find ways in which we start to communicate the regulatory mechanisms and framework. That way people will appreciate, they will then appreciate that in actual fact, what they are eating or what is already out there in the public that has never gone through such rigorous regulations is perhaps even more risky than what we are developing. So yeah, and I think as SCICOM, we might want to pick up this role and build awareness around regulation, how exactly it is done, who does it, for what purpose, and therefore ensure that people value and trust the product that we deliver.
Okay, I agree with you on that, Andrew. As SCICOM, that's a mandate that we can pick up as well. So Murenga, any comments before I move on to the next person?
No.
Okay, so I can see Joyce, your hand is up. So you have the floor, Joyce. Go ahead.
Unmute.
Joyce?
Yes, you're muted.
Okay. Hello, can you hear me?
Yes, very well.
Okay, Joyce Jechi is the name, and I'm a journalist. I happened to be on a tour to Abuja 2 years ago when they did a demo on it as, yeah, like taste and see. Yeah, that was what they called it at that time. And that had to do with the GM cowpea. So during that interaction with officials and the farmers and all, we got to know that there are science research institutions like what we have in Ghana at the Council for Scientific and Industrial Research has a radio station. So this is a science, a research institution that has a radio station to really spread or put out the message of the successes and all that the biotech is, the technology is about. In Ghana, we have the police service now has a television station that they put their message on. My question to the panelists is that they should share their experience with what pertains in Kenya. Does Kenya also have a station, or other, let me say, other countries have radio stations pertaining to research institutions apart from Nigeria that I happen to be there to see in spreading out the message? I would say if the researchers really put out their message across, this is what we've seen, and keep educating the people, I think the message would be accepted. So apart from Nigeria, what other country has a research institution that has a radio station? Thank you.
That's a very interesting observation. And Kenya, Andrew?
Yeah, yeah, a quick one that doesn't help very much. In fact, radio extension has been around for some time. And if I remember, the National Culture Research Organization in Uganda did have at least 3 radio stations, community radio stations. But for some reason, I think I also remember one in Tanzania and other places. So it's not new, it's been around, but I think this is something we can bring back. Something that we can bring back. They were abandoned. I don't know for what reason.
I would want to say something that in Kenya, we don't have dedicated radio stations coming from research organizations, be they agricultural, health, or otherwise. But we have national radio and other private radio stations that make a broadcast in vernacular languages. So the outreach, I mean, the level of reaching out to the public, even with vernacular TV stations, be they private or other or public, is that they are available in Kenya, and there are very many, even at the county levels. These are like regional radio stations, in vernacular stations, including TV. However, my little experience with stations, be they TV or radio, and particularly those that are vernacular. One of the biggest challenge that we have is managing the expectations of the people. For example, with the BT maize that was approved in Kenya for uptake recently, the question is, you, the research team, and the rest of the people go to the vernacular stations and make announcements that this maize is good in this form, in this form, in this form. And you fail to manage the expectations. In this case, you're creating demand. And while the expectations are not managed, in this case, would mean that the farmers, immediately after the switch of the radio, they go to the market and ask for this seed. And yet, the research team has not put in place the seed. The seed is not on the shelf. So an equivalent of the same is that while we talk about gene drives and GM mosquitoes and the efficaciousness of its effectiveness in terms of controlling malaria in those target environments, we must be very careful on how to manage the expectations of the people. That is my take.
Okay, thank you so much, Dr. Wimali. I see all the hands are down. We had a hand up. But yeah, so Christopher, I will allow you to give us the last comment for the webinar. So please go ahead.
Yes, I just wanted to follow up and say that, you see, in Zambia, we do not have a research institution per se that has a radio station, but we have National Agriculture Information Services and the public radio stations as well as private stations. But that does not deter us to make sure that we have airtime and we can publicize and talk about anything that we are doing in relation to regulation of genetically modified organisms, in relation to people that are trying to innovate in as far as the biotechnology or genetic engineering is concerned. I think it's just an inertia that has been created by the GMO activists and the sentiments that come out that we are not managing to defeat. So I think it is time that we went out and began to make sure we give the people the right information. You see, propaganda seems to take toll. Of course, they are paid, like I said, what colleagues said there. They are paid, and so they want to make sure they achieve what they want to achieve with the agenda that they have. So I think scientists and the journalists, it's time we went out and make sure that we give the people the right information. And you see, if like what is happening in Kenya, here is a seed that is BT, and it's a variety that has come, it's a good variety, and they can't find it in the market. It's processes, you see. Maybe they are yet to make sure it's commercialized so that it's ready for the market. But all in all, what I'm trying to advocate for is that we just need to make sure we speak out the right information to the public, and the public will be able to take the information and uphold it correctly, other than propaganda that is circulating all over. Thank you.
Okay, thank you, Christopher, for your comment. So I want to bring the webinar to a close and thank everybody who has participated here today. And I can see we had good representations from the various countries in Africa. We had Tanzania, Burkina Faso, we had Nigeria, we had Ghana, Zambia, we also had Kenya and Uganda. So I want to thank everyone who participated and joined today. Thank you for your time, for your comments, for your questions. And to our speakers, Andrew and Dr. Murenga, thank you so much for the information and the presentations that you have educated the team today with. So for the next webinar that's coming up, we have a 4-part series webinar. This is just our first. The second one is on the March of 14th. And in the next webinar, we are talking about ways of addressing and communicating the uncertainties associated with emerging biotechnologies. And here we'll be looking at the role of communication in genetic biotechnologies, and we'll also be looking at the impact of communicating genetic biocontrol technologies in Africa, some of the stories and case studies by journalists and scientists in Africa. So I look forward to seeing the team and more, and we hope also that we will be able to get some good, amazing stories from these webinars as well. So thank you everyone for participating. So Aruna, I can see your hand is up. Let me give you just 1 or 2 minutes to talk, and then we can close this up. So Aruna, you have the floor. Aruna, please go ahead. Okay, so it seems like Aruna is not talking. So please allow me to wrap up the webinar, and I wish all of you a good rest of the day, a good evening, and a good day, and a good night as well.
Bye.
Okay, see you everyone. See you
Yeah,
on the
bye.
14th of
See
March.
you all.
Okay.
Bye, thank you, and thank you all for your participation.